I play that. Okay, so I think we can. Okay. Good afternoon, everybody, and welcome to the departmental seminar of CBS department. And it's a pleasure to have Dr. Soumya from IIT Kharagpur. We go a long way back. He's a friend, and also have started collaborating recently. So Soumya did his bachelor's and MSc in chemistry, I think, from IIT Kharagpur, and then he did PhD from Cornell University. Then he was a postdoctoral researcher in University of British Columbia in Canada. He came back to India in 2015, joined IIT Kharagpur as faculty, and he's a structural biologist, but he uses solution NMR as a technique. So beyond structure, he also can look into dynamics, and that's where we overlap a lot. And he's interested to look into the structure and dynamics of both disordered and ordered proteins. But today, I think he's going to talk about mostly on the disordered proteins. So.
Thank you, Suman, for the kind introduction and also inviting me here. And thank you all of you for coming to the talk. Right. So, as Suman said, that my lab is a structural biology lab, and I use NMR spectroscopy to look at protein structures. So, in the past few years, we have solved structures like this. Right? So, this is typically what we structural biologists do. But beyond that, we also started looking at proteins which have these long disordered regions, right? And I'll show you what I mean by that. So if you think about the spectrum of protein structures, right? On one end, we have proteins like this, which are completely structured. So one example will be this protein. This is an enzyme, and it is structured right from the first residue to the last residue, right? On the other end, we have proteins like this, which are completely disordered, right? So their examples are P27, alpha synuclein. So there are these proteins which are completely disordered. There is no secondary structure. There is no tertiary structure. Right? And in between, you can have proteins like this, where in this case you have a folded domain and then a long disordered region. Right? You can have multiple domains connected by linkers and some disordered regions, things like that. So these are different spectrum types of proteins that we see. So in the jargon of intrinsically disordered proteins, we will sort of club all of them as IDPs or intrinsically disordered proteins if these disordered regions are more than 30 residues long. So that's sort of the typical definition in the field, that if you have consecutive 30 residues which are disordered, then you can include it in this class of intrinsically disordered proteins. Okay. And these proteins are quite abundant. So you can actually find them in all 4 domains of life. So you can find them in bacteria, eukaryotes, archaea, and viruses. And you will see that we find proteins with longer disordered regions or more proteins with disordered regions more in eukaryotes compared to bacteria. And one of the reasons for that is we get more protein-protein interactions in this eukaryotic systems. And these disordered proteins, they act as hub proteins where they can interact with multiple proteins. So we tend to see more disordered regions in eukaryotes. And virus is an interesting case because depending on what type of virus you're looking at, either it will mimic the eukaryotes or it will mimic the prokaryotes, right? So if you look at bacteriophages, then they will be more like this. If you look at viruses which infect us, they will have more disordered regions like this. So if we think about the functions, how these disordered proteins function or what exactly they do, we can sort of divide them into these 4 groups. So as I said that these are disordered, so they cannot actually have catalytic functions, right? So what they mostly do is they act as interactors with other proteins. So disordered proteins are found a lot in multi-protein complexes, right? So they sort of facilitate these protein-protein interactions. They are also sites of post-translational modifications like phosphorylation, glycosylation, and things like that. They, just like proteins, they can also interact with nucleic acids. So there are proteins which bind with DNA, there are regions which bind with RNA. They can also act as autoinhibitory elements. I will show you examples of this and this. And one thing that is not listed here is that these disordered regions, they can also facilitate phase separation or formation of biomolecular condensates. Right? And this type of disorder is spread throughout the cell, right? So if you think of a typical eukaryotic cell, we find them in the nuclear pores, we find them in histones where epigenetic modifications happen, we find them in these membrane proteins, for example, in GPCRs, where these disorder regions are the ones which help in the modeling or controlling the function of the protein. They help in the formation of complexes. Um, I will mostly talk about this. So disordered regions are found a lot in transcription factors. So in my lab, we actually look at transcription factors. So these are proteins which regulate the expression of different genes, right? And these transcription factors, they tend to have long disordered regions. And not surprisingly, these proteins are also found in many diseases. So some of the typical diseases are sort of listed here, like cancer, diabetes, cardiovascular diseases, neurodegenerative diseases, right? So you can see that the percentage of proteins with different lengths of disordered regions, they are listed here. So we actually see a lot of disordered proteins with very long disordered regions, which are involved in different types of disease. Now, what is the difference between a disordered region or an intrinsically disordered protein versus a folded protein? So why some sequences fold versus some sequences do not fold? Right? And if you think of Robinson's experiment or the thermodynamic principle of protein folding, then we should be able to tell just by looking at the sequence of a protein whether it should fold or not. And that is exactly the case. So quite some time back, Uversky, he actually showed that if you calculate the mean hydrophobicity of a protein and the mean net charge of a protein, and you plot them, we can actually see a distinction between proteins which are disordered versus proteins which are natively folded, right? And this line which separates these two is called the Uversky line. Okay. And this is sort of you can intuitively you can see that yes, this is expected. Proteins which will fold, they should have more hydrophobic residues because they will have this hydrophobic collapse. So their mean hydrophobicity will be higher. And proteins which are disordered, they will lack those and they will be more soluble. So you will have more polar groups. So their mean hydrophobicity should be less. On the other hand, if you think about the net charge, proteins which have a higher net charge, they will be more soluble. And proteins which fold, they will have a lower net charge. So this is actually a very good way of distinguishing between folded and disordered sequences. But this was like almost 20 years back. Now there are many programs where you can put the sequence and they can tell you which part of the sequence is folded, which part of the sequence is disordered. Right? So just by residue wise, you can actually get that information.
Clarify. So this distinction is based on the aqueous medium. So
yes.
If you change the environment or give it something to bind to, so it can adopt other structure, even another environment.
Right. So this is the free protein.
Somehow
yes.
that is also encoded in the disordered.
Yes. Yes. So this one is just for the free protein in the absence of any interactions, and that happens. So that is the major challenge, that in the cell these proteins are not free, right? And we will see one example of that. Okay. So since these proteins do not have any, so maybe I should say that these proteins sort of look like this. So alpha synuclein, it's a completely disordered protein. So it is shown here, but you can see that it samples all these different conformations. One is shown in blue, one is shown in red, right? And then all these different gray ones, right? It's true for other proteins also, which will have a folded domain with a disordered region. So for example, this one, that's the folded domain, and you see the disordered part is sort of flopping around, right? And this degree of disorderedness is also something that can vary. So again, if we look at disordered protein or just any protein, you can actually see that the degree of disorderedness can also vary. On one end, you have this completely folded proteins. On the other hand, you have this completely unfolded protein. So this will be an extended conformation, right? You can have something like this where the protein forms transient secondary structure. So you see a small alpha helix has formed here, or sometimes we see a small beta hairpin forms, right? So those transient interactions happen and it forms this transient secondary structure, but then again it opens up because there are no tertiary contacts which stabilizes those secondary structures. Even without formation of any transient secondary structures, it can sort of form a bit more of a collapsed structure. So this will be a compact globule. This is a molten globule where you have secondary structures, but the tertiary contacts are missing. And this is more of a folded protein with some big loop, and this is a completely folded protein, right? So for disordered proteins, we actually see anything, things that can sort of sample these different conformations. And that will also drive what people have actually observed, that if there are regions which transiently form secondary structures like that, those tend to be the parts which will interact with other proteins. So that's sort of a preformed state for the bound complex. So energetically, we can think of it like this, that folded proteins, they have this much deeper folding funnel. So it starts somewhere here and then it will fold into this nice native structure. But for the disordered proteins, the folding funnel is very shallow and it can hop between all these different local minima where these are different conformations. So this is the one of the proteins that we work with, and here you can also see that there is a folded domain which stays very stable, but the connected disordered part, it is all over the place. So, and that brings the challenge for studying this type of disordered regions or disordered proteins, that this looks a very chaotic system. Right? So here I'm using the word chaos because title of my talk is functionality of chaos, right? And so what I want to do is just spend one slide on this description of chaos, right? So it's actually a proper physical term, and so examples of chaotic systems, a very prominent example will be weather. So if you want to forecast weather, it is very difficult because weather is a chaotic system, right? Share market indices, our heartbeat, irregularities in our heartbeat, the formation of blood vessels in different organs. So it's basically follows a fractal pattern, which is also a chaotic system. So these are some examples. There are many, many examples in biological systems also. Now, these chaotic systems, they are not random systems. Okay, so normally we associate the term randomness with chaos, but that is not what it is. These are actually deterministic systems which can be defined by a set of nonlinear differential equations. You can precisely, if you know the initial condition, you can precisely predict what will be the condition after a certain amount of time. Right? But the problem is that these systems are dependent on this initial system, and if there is a little bit of change in the initial system, very quickly these systems can diverge. So you can have very wide differences in the predictions depending on small differences in the initial system, which makes these systems almost impossible to predict in terms of their long-term behavior. Okay. So very good example of a chaotic system will be a double pendulum. Right? So normally we have seen a single pendulum where you have, let's say, a string and you attach a big wall and you can oscillate it. Right? So we can write equations and we can predict exactly where it will be at a certain point of time if we know the initial condition. And small changes will not make a big difference in your prediction. But in case of a double pendulum, let's just wait for it to reset. So this is a double pendulum. So there are two hinges, right? And you start with some initial condition where three pendulums, they are taken at a certain point and you let it go. Okay, so you can imagine that the initial conditions are very similar. You can see it starts like this and then quickly it is diverging. You see the tip is now completely different, right? So it's sampling different x and z coordinate within 3 seconds. So in 3 seconds, it diverges into something very different. So that's the long-term prediction. Okay. There are many examples. I mean, this is most probably the most famous simulation for chaotic systems, which was actually done by Edward Lorenz, and this is called the Lorenz attractor. So if you take any 2 points and start them, you can see that after some time they will sample completely different parts of this phase space. Right? And this is Lorenz. He had this very cheeky title of his paper when he presented it. It's called "Does the flap of a butterfly's wings in Brazil take off a tornado in Texas?" And this is famously called the butterfly effect. Right? So very small changes here can have very big differences somewhere else. Right? So why am I talking about this? Right? The whole point of this thing is that what I'm trying to tell you is most probably intrinsically disordered proteins represent a chaotic system. Right? So not folded proteins, because folded proteins go through this funnel and when it reaches there, it has a native state. Right? But disordered proteins, they sample all these different conformations, and people have actually seen by simulating small peptides that you can start with initial conditions, and within few nanoseconds, the conformations become very different. So there is a particular parameter called the Lupinov number, which is sort of an exponential factor, and within 10 nanoseconds, you actually get this divergence for small peptides, right? So it seems like these disordered sequences are an example of a chaotic system. But then that makes it difficult for us that how do you study something where the long-term behavior is difficult to predict. So there are several ways people have started doing experimental experiments on disordered proteins. One is that since we are structural biologists, what we do is we try to solve structure, right? So the initial thought was that let's try to solve all these different structures, right? So if I can solve a big ensemble, because we have to think in terms of ensemble, there is not a single structure, there are multiple structures. If I can solve an ensemble of structure, maybe I can predict the behavior or explain the behavior of this protein. But then the problem was that how many structures are you going to solve? Is it 100? Is it 1,000? It is 1 million? It seems like it has to be in the order of several million, right? And solving that many structures is quite problematic. And what are the restraints that you are going to use? Another way of looking at it is don't think in terms of structure, think in terms of dynamics, because these are highly dynamic systems. And that is what we sort of try to do in my lab. So I'll just introduce the systems. I'm not going to talk about this one. I'll just briefly introduce it, but I will focus mostly on the transcription factors. So one set of proteins that we work with are Hox transcription factors. So these are roughly 300 amino acids long. Only 50 residues are folded. The rest is intrinsically disordered. And what we have seen, at least in the example of these proteins, is that these disordered regions, they have small segments in them which act as hotspots for protein-protein interaction. So they recruit different proteins. Another example that we have seen is part of these proteins folds back and prevents the folded domain from doing its function. So basically its function is to bind DNA, so it prevents the DNA binding domain to bind DNA, and that is how it regulates its function. Third example that we have seen, this is not exactly on Hox protein, but on one of the partner proteins, is that there is a disordered segment towards its C terminus. When this protein binds DNA, it folds. Right? So in the free protein, like someone was saying, it's actually disordered, but when it binds DNA, this folds. But interestingly, this folded part, it forms a new fourth helix, and it is not interacting the DNA. Okay, so some subtle changes happen in the protein where this disordered part becomes completely folded. This other so these are DNA binding proteins. The other protein that we are working with, it is an RNA binding protein. It has a folded N-terminal domain. We have solved the structure of this by NMR, and it has a long disordered region towards the C terminus, and it actually forms phase separation. So just like oil in water, if you make this protein, it separates out into small droplets, and we can actually see it under microscope. But I'm not going to talk about this today. And this type of droplets or membraneless organelles are something that are found throughout cells, and they have different functions. Okay. So what is this protein, Hox protein? In human, we have 39 Hox proteins, and they are separated in these 4 different chromosomes. And typically, what they do is they're responsible for the body plan of bilateral vertebrates. Okay, so we are bilateral, we are bilaterally symmetrical. So this type of animals, their body plan is defined by this Hox protein. So these proteins will define these legs and this lower part, abdominal part, and these proteins will define where the head will be, where the vein formation will happen. So all of these things are defined by that. But that's during developmental stage. After development, these proteins are involved in other functions. The one that we are looking at is the formation of blood. Okay. So they are involved in different blood formation, and when their function is not working, of course, it causes different types of disease, including leukemia. So these are some of the proteins that we are working with. So 3 from human and 2 from Drosophila. The folded domain looks like this. It's a typical homeodomain. It has 3 alpha helices. The third helix inserts into the DNA major group like this. And the disordered part, it interacts with the partner protein. So together, these proteins work. They bind different parts of the DNA, and then they recruit different factors like the co-activators or co-repressors. Right. So, what type of partner protein is recruited here by this protein will determine whether co-activators are recruited or co-repressors are recruited, and that will determine what happens to the downstream gene, whether it is expressed or whether it is repressed. I'm not going to go through all of this. It's just
the partner protein is sitting on the DNA.
Yes, the partner protein also has a DNA binding protein. So, as I said that instead of looking at structures of these disordered sequences, we are more interested in looking at the dynamics. And NMR spectroscopy is something that is very useful because we can sample all these different motions by different NMR experiments. A single experiment cannot look at all the motions, but there are different experiments which can look at these different motions. And since it's we are looking at disordered part, this type of motions happen like torsion angle dynamics, fast loop motions, side chain rotations, things like that, which happen in picosecond, nanosecond time scale. So we normally use this R1, R2, and NOE, these experiments which report on this type of motions. So I'm not going to show much of NMR data. This is sort of a summary of what we have done. So using this type of R1, R2, NOE experiments, what we can do is we can calculate something called spectral density. So this is nothing but it tells you so this is per residue. Okay, so we have this 90 amino acid protein, and we can look at every residue, and we are looking at the backbone dynamics, right? So we can see, so these are slow motions, these are fast motions, these are sort of intermediate motions. So you can see that these residues have less amount of fast motions and more of slow motion, and they turn out to be the folded domain. These residues, they have more contribution from the fast motions and less from the slower motions, and these are the disordered, which is expected. The folded domain will tumble slowly, it will have slower motions, more of slower motions, and the disordered part will have more of the faster motions, right? And if we zoom into this, we can see that even in the disordered region, you can actually see this variation, and we can pick up residues which have more slower motions and less faster motions, and residues which are more faster motions and less slower motions. So even in the disordered region, we can see there are some residues which are tumbling slowly and some residues which are tumbling faster, right? And then we have shown that, okay, we can use combination of these three and sort of translate it back into the original.
When you say faster and slower, what kind of time scales are you indicating?
Yes, so typically this will be in terms in around 10 nanoseconds, and this will be in the range of 50 to 100 picoseconds, and this will be around 1 minute. So that's roughly that. So what we have shown that we can actually use this R1, R2, and NOE values to.
So are these two related? Meaning what you showed is when it is slow, or you're only looking at back?
Yes, we are only looking at backbone. Yes, we are only looking at backbone because the side chains will be anyway faster for this. Yes, so I mean, we first did all of this analysis to pick this up, and then later on we showed that you don't actually have to do all of this analysis. We can just from the actual NMR parameters, we can show by using something like this, where we multiply 2 and divide by the third one, that we can pick up rigid segments, residues which are more rigid compared to the rest of the protein, right? So based on that, I mean, this thing was already sort of known, and I think this data supports that these disordered regions, you can think of them as rigid, having rigid segments. So this is a disordered region. These few residues, so around 6 residues, they form a rigid segment, and then there is a flexible region, and then you have another rigid segment. So you can think of it like this, my hand, right? So there is a flexible region here, and there are rigid segments here. So these rigid segments are connected by these flexible regions. So we can do that by NMR. Okay, that's fine. But then the question is there any biological significance of rigid segments like this? So what we did was we took this protein, and we know that the partner protein actually interacts with this protein, and it interacts with the disordered region somewhere, right? So we did a titration, and in NMR we can actually look at residue-wise effect. So what we do is we label one protein and the other one is unlabeled, and if you titrate it, you can see exactly which residues are interacting with the other proteins. So we did experiment like that. So we have this as labeled, and this one is the partner protein is unlabeled. So these are chemical shifts which we are plotting, and that's all the residues, right? So you can see that most of these residues are not interacting. Very small chemical shift perturbation. So that's basically noise. And there are these 6 residues. We don't see one because that's a proline. Proline does not have an NH group, so we don't see that in this experiment. So the remaining 5, they have this very high chemical shift perturbation. And these 6 residues matches exactly with these 6 residues. 1, 2, 3, 4, 5, 6. Right. So there is one. So it turns out that this rigid segment, which sort of maintains this rigidity in the free protein, is the one which is also interacting with the partner protein. So we did this experiment without any DNA. Okay. So even without DNA, these 2 proteins can interact, and the interaction is highly specific for this rigid segment. And that is one of the characteristics of disorder proteins, that the interaction between proteins is highly specific.
In the previous slide, what about the rigid section? I cannot see
this one. Yes, yes. So I did not talk about that. This one interacts with DNA. So you see, when it looks over like this part is one that interacts with the partner protein, and this part is somewhere here, and it interacts with the DNA. Yes.
So in that part, 6 residues that interact. Due to the sequence, I see W, P, Y.
Yes, yes. So it is
so proline is on both sides you have aromatic. So looks like what did you see as cis or trans, or did you see?
So in HSPC we see 2 things. There is a cis population, but the one that interacts is the trans.
You know, can you tell me how much is 2030 almost per person?
We can talk it. I don't remember, but I think it's. I mean, if you look at the intensity, I think it is somewhere around. Yeah, maybe less than 20%. So maybe between
the free peptide YPW will be about 20. But anyway.
So yes, the cis is there. So cis is there.
Yeah, in the folder it has to choose either cis or trans.
Here there is no constraint. But when it is interacting, it has to choose. Okay. So all of this work was actually done by the Hox proteins taken from Drosophila, and so the next one that I will show you is for this Hox protein, which is taken from humans, and it is called Hox A9. So in this case, what we did was we actually have not the full length protein, but we made a protein which is this long, because one of the challenges of this disorder protein is that they degrade very fast. So we are still not able to make the full length protein, but this is what we can actually get, 101 to 270. And the first thing that we did was we wanted to look at its interaction with DNA, promoter site. To do that, to systematically look at it, what we did was we had this construct, and then we made truncations. So 151 to this, 194 to this, and then this minimal homeodomain, right? And then we tested its DNA binding. One of the interesting things that we saw is that if we have only the homeodomain, it binds DNA with 42 nanomolar, which is quite nice. But the moment you introduce these disordered regions, the affinity goes down. So the KD value goes up to around 240 nanomolar. So this is almost 6-fold inhibition, right? And you can see that irrespective of the length, these values are very similar. So we reason that most probably something in this minimal construct, so something here is happening which is inhibiting its DNA binding function. So, and in NMR, we actually do experiments like this. So what we do is we make it as N15 level, and you can look at cross peaks like this. So each of these cross peaks represents the backbone image. You can also see some of the side chains here, but each of these look correspond to the backbone image here. So if I look at this peak, let's say that's the proton chemical shift, and this is the nitrogen chemical shift. And then we use 3-dimensional experiments to assign each of these and know exactly which amino acid, which peak belongs to which amino acid. Right? Once we have that, we can do lots of experiments. So the first thing that we did was we looked at its structural integrity, and we see from the chemical shift of the backbone carbon, C alpha and C beta, that this part is indeed folded. You see, we can actually see 3 helices formed, and this N-terminal part is disordered. Then we did this favorite experiments of R1, R2, and NOE, and you can see that again this part is folded, so we get positive NOE values, and as we get into this region, it drops and goes to negative. So that is very indicative of fast motions, which means it is integrally disordered. So that confirms that yes, this part is disordered, but it still doesn't tell you what it is doing to inhibit the DNA binding. So for that, we actually did a little bit of MD simulation of this construct. So we had a 1 microsecond MD simulation, and there we can see. So this is the radius of gyration plotted for the protein. You can see that the radius of gyration is high, then it comes down, then again it goes up, and then again it comes down. And if you take snapshots at different time points, you can see that here it is extended, and then it collapses, then again it goes back out, and then again it collapses. And if you look at these structures, you can see that there is a tryptophan which interacts with this domain. Actually, not just that tryptophan, there are residues around that tryptophan which contact this protein. So there are pi-pi interactions. There are so pi-pi interaction is sort of seen here. There is charge pi interaction. So arginine is interacting with this tryptophan side chain. So all sorts of interactions we actually see, right? So based on that, we propose that okay, most probably this
part of that WPY.
Yeah, so it turns out to be that same tryptophan. Now the problem with disordered proteins is that they are not highly conserved. So in this case, it is YPWM. But in this case, it is just only W. It is preceded by alanine, followed by it is A and W something. So the sequence changes. There is only one W. So based on one W, you cannot tell it's the same thing, and also the spacing changes, right? So sequence alignment is very problematic. But when you do experiments like this, you can tell that okay, most probably this W and that W are same, and that is exactly what we see.
So in your MD, your WP was trans.
Yes, but here there is no P. There is no P. So that P was for this Drosophila protein.
I see. So P has gone.
Yes, P has gone in this case. So Y, P, M, everything is gone. This W is preceded by alanine and aspirin and something. So, but W is there. So we suspected that okay, most probably this W is one of the residues which is driving this sort of interaction, and most probably it is inhibiting the DNA binding. So the simplest experiment is to mutate that and then do the DNA binding experiments again. So when we do that, so this is this small construct, right? And it is shown here again. And these ones are these constructs with the W mutated to alanine. So when we mutate it, it actually comes down. And this is very similar to this, just a minimal conversion, which means that once we mutate that W to alanine, this interaction is not stabilized anymore. So it is not doing this much, or at least not staying here much. So the transient interaction is gone.
And so 6-fold change means in KD, it means minus RT ln K1 is 1, L minus R. So I think that turns out to be how much kilocalorie roughly you can calculate, and then you can think of the interaction that W is having. Right? Maybe so only 1.2 or
6.4 around 1 thing else.
So now whatever interaction you are looking at in your end, you can guess what the rest of what. I'm just saying.
Yes, yes, that's a good point. So yeah, we can actually do that because we have the
back of the envelope.
So yeah, that's, I mean, even though it's a transient interaction, it still makes that difference. So it turns out that that tryptophan is the one which interacts with the partner protein. So when the partner is present, it will actually interact with this, recruit that partner protein, and together they bind DNA. So we are actually doing this experiment now, and we can see this cooperative binding. So you have the partner protein, the DNA binding becomes better.
So in the previous slide, it seemed like it's tryptophan.
So
stacking.
Yes, there is a tryptophan, and there is also tryptophan arginine interaction because there are lots of arguments.
Okay, so cation pi interaction, pi interactions.
But still, these are very transient interactions, not stabilizing it.
No, that data of changing your KD is very. It's too good to be true. Look like you're
Yes. So that's why we did it for all the three constants, just to make sure that it's. Yeah. So this is the model that we have for this protein. That in the presence of the partner protein, this tryptophan, it actually has this dual role, or the disorder region has this dual role. In the presence of partner protein, it helps both to bind DNA cooperatively, and together the binding affinity actually goes up, becomes much tighter than 40 nanomolar. But in the absence of partner protein, it inhibits DNA binding. So in this case, this is around 240 nanomolar, and in this case, it will be much tighter than 40 nanomolar. So depending on whether the partner protein is present or not, it will drive it in this direction. So we also looked at the partner protein, which is this extradentically, and its structure is also, so that's the partner protein, right? So normally they interact like this. So this is the Hox protein, this is the partner protein. The disorder region loops over the DNA, and so the second rigid segment that you are asking about, it's here, and it sort of interacts with the partner group of them. And then the other one, it interacts with the partner. So we looked at this partner protein. It also looks like this because it's also a homeodomain, and its helix 3 inserts into the DNA major group like this. Now in this case, so there are 3 helices, and this is the C-terminal part which is intrinsically disordered. So by NMR we can actually tell that. Right? So we have done this R1, R2 NOE experiments. We can tell it is disordered, but it is not as disordered like this. So I did not show. I don't have the data here. So here you see that disordered part becomes negative, but in that case they are slightly positive. It means that they are not as flexible as this disordered part. So this C-terminal tail is not as flexible as that. Now, when we add DNA, it folds into this 4th helix. It's a really nicely formed, and it's quite big. It has 5 terms. Okay. But interestingly, you see there is no contact between this 4th helix and the DNA. So the 4th helix is not contacting the DNA at all. But somehow, binding of this to the DNA stabilizes this 4th helix. So it makes some small changes in the structure of the protein, and it forms this. So right now we are trying to solve the structure of this.
Yes, you have the answer. You have the structure for this one.
No, we are trying to get this. So we are trying to get the free protein structure and as well as the DNA bound structure.
The other thing that you may want to look at is electrostatic effects, long-range electrostatic effect from DNA. So, very subtle, but helix dipoles play a role.
Right, right.
If it forms a dipole, then
Right.
it becomes favorable in the electric field. The DNA is very charged.
Right. So that is what you said is absolutely right, because this is your N terminus, so that's the positive part of the dipole, and that is most probably a big dipole.
It doesn't need much energy, but that's just enough to move it to a helix.
So, so what we are trying to do is we are sort of trying to get this again. This is something that we are trying to figure out is what is sort of the thermodynamics of this binding induced protein, right? So the model that we have built, or this is something that we are trying to get, is so this is the free protein, but the free protein you can think of it as something that is exchanging between these two states, where this is completely disordered and where it is actually folded into this helix. But the population of this is much less than this. So maybe this is, let's say, 1%, this is 99%. Right? This part can bind to DNA, and this is the state where this is bound to DNA. This conformation can also bind to DNA. So now it is bound to DNA. And these two conformations can also exchange among each other. Right? So this is basically the thermodynamic cycle of this protein binding to DNA. And if you think about the classical binding models, then this part will be the induced fit model, where binding of the protein induces its folding. So that's the induced fit. And this part will be the conformational selection model, because you have these two conformations, and this one is selected and it is being bound by DNA. So ideally, the thermodynamic cycle is actually a combination of both, and we can tell which of these two paths is followed if we can get all these equilibrium constants and all the kinetics. So we have to get all the kinetic rates of all the steps and all the equilibrium. So then we can actually tell if this is the major pathway or the major pathway. And of course, we can also get more insights into what drives this folding of the. So for that, what we are doing is this one is easy to get. All you do is chop off this theta middle part. So we have done this. We know the KB of this. Now we are doing SPR experiments to get the kinetic rates. We are also planning to do CPMG to get this exchange of conformations and the relative population and the rates of exchange. This part is tricky. So for that, what we have done is we have designed some mutations which stabilizes the helix. And those mutations are actually far from the DNA binding site. So what we have done is we have driven the population to this side. So from NMR, we can tell that it is more like this, right? And now we are trying to get these KD values at this kind. So once we have all these different equilibrium values, we can then define thermodynamic cycle in a better way.
So whatever you get will be between conformational selection and in lead state. Partly this, partly that, and the ratio will depend upon the concentration. Right? Because it's at the end, it's a bimolecular reaction.
Right. So it will depend on the the flux will depend on the concentration.
There's a paper in PNS. Are you're aware of that, right? I think Gordon Thomas is there.
Yes, it's old paper.
But that is what?
That is exactly what they showed. That depending on the concentration, your flux actually changes from this to this. So
yeah, and often I think when we look at biological reactions to it, we forget that for bimolecular reactions, there can be local increase of one of the reactants. Then it's going to shift. Anyway, you would know this. Very
yes. Yeah, so for that, ideally, we should have this instead of just having one or the other monade constant. This is the full. So I think that is all I wanted to talk about. So, so that's sort of a summary of what I was trying to say. Is that intrinsically disordered proteins are something that we should not ignore. Traditionally, we structural biologists, what we would do is just chop off the disordered parts because these are nuisance, and then look at the structured part. But now I think we have the tools, both experimental and computational tools, to study these very interesting proteins. And we have looked at Hobbs transcription factors, and we see that they provide this functional specificity to these proteins. Because if you take these 39 Hobbs proteins, if you look at only the folded domain, they are 80% identical. Okay. So the functional specificity actually comes from the disordered part of these proteins. Even in the disordered part, we have seen that they are not completely disordered to the same extent. Some parts are more flexible, some parts are more rigid, and we think that the parts, the small segments which are more rigid, they are functionally important, and they most probably constitute what we call short linear motifs in IDPs or SLIPs. So these selective rigid regions can selectively bind to partner proteins. They can also be hotspots for post-translational modifications. Disordered regions can also modulate protein-protein, protein-nucleic acid interactions. We have seen it in the context of DNA binding, that we have seen that there is autoinhibition in one case, and in the other case, we have seen folding upon binding. So one important thing that I forgot to mention is that when we get this KD, it is something in the range of around 140 nanomolar when we cut off this disordered part. But when the disordered part is present, so basically we are going from this state to this state, the affinity is actually tighter, threefold tighter. And which I think is sort of counterintuitive, because in this case, what happens is that you are, so there is an entropic cost, right? Because we are folding this, right? But still the affinity is increasing. So that's also one thing that we are trying to figure out. And most probably this dipole interaction is most probably. So I like to work with lots of people. So there are my collaborators within the campus, and also there are collaborators like someone in other parts, and we get funding from these places, and most of these works are actually done by these students. So some of them have actually graduated. So Snigdha and Vidisha, they did the work on the Drosophila hops proteins. Nikita, she did the work on extradentical. Vidisha initiated the extradentical work. Sorry, Nikita did the work on COX-A9. Vidisha, she initiated the extradentical work, and now it is being continued by others. And thank you.
Thank you so much for fascinating talk. Questions?
Yes.
Sir, I have a couple of questions. Thank you for some. So, sir, in one of the slides you showed that there is a plot which actually defines the intrinsically disordered proteins and the folded protein. That plot. Yes. So now, sir, if I think in terms of engineering a protein, where we want to increase the solubility of the protein.
Right.
So in that case, if I want to increase the solubility of the protein, I have to compromise the hydrophobicity of the protein. So in that case, how can I tackle that problem in order to, it should not go to the intrinsically disordered region, and also it maintains its mode and increase the solubility.
Excellent. So yes, so normally, I mean, if you already have a folded protein, right, then part of your job is already done, because these folded proteins are for the hydrophobic residues are not randomly present, right? So they are present in such a way that they form this hydrophobic core. So in that, if you know the structure, what you are going to do is you are going to target only the surface residue. So you will try to mutate. Let's say you have a leucine, which is surface exposed, and you see that it is not making much contact. So you can mutate that to lysine or something like that, and that will improve your solution.
The surface.
So yes, you will of course target the surface residue, but that will you can only do if you know the structure of the protein. But now with AlphaFold and things like that, I mean, predicting the structure of structured proteins is sort of is quite good. That is what I.
Yeah, the important question is, do you know if your protein is a monomer or a dimer or not? You also have to know that.
That will
Because you follow sir's suggestion,
then you will be in
and
trouble.
change a hydrophobic to something else, and this is how hemoglobin 1 value changes to glutamate, and you get sequence variant. So what he said is, once you know the structure, then you have to think whether this, why is it not so? Maybe because it's forming aggregate. Maybe something is happening. So your PhD student either change your problem or change your problem. And your advisor will never tell you which one to do. I had a quick, you have more questions?
Yeah. At least another question.
Sir, sir, in the plots, when we are when we are visualizing the motions, the motions of the residues, in that case, while capturing those motions, are we taking any external perturbations, environmental, external?
So it's
That residue wise, yes.
this one.
Yes. Are we taking the external environmental perturbations as suggested by sir also?
So environmental perturbation means this. See, these are in vitro experiments, right? So what we have is you have the protein in some buffer, right? And that is what you are looking at. So there is no partner protein or so by environment. If you think about if you think about other proteins or in cell environment, then that is not there.
I'm
is just a
thinking in terms of water, role of water.
Yes, yes, water is there because this is in some buffer.
So how much this water can fluctuate the thermal pressures of the residues in that case? Will it capture this particular technique?
He's measuring the actual dynamics of the protein in presence of everything. Whatever is happening is measuring the movement.
So water, salt, ions, everything is there. That way, it's a complete setup.
That's the last question.
Later, maybe we come back. We'll come back.
I had a quick thing. So can you go back to the previous? Yeah.
Oh, this one.
So, yeah, whatever. Here you have shown regions of proteins that are intermediate in dynamics, more dynamic as time. So a very simple way of also looking at it, but slightly different, is to take your protein and digest it in native under native condition and do mass spec. Do you also sometimes do and corroborate and
Yes. So yeah, we do limited proteinases. So we take the protein and we add non-specific proteases to see whether a part is intrinsically disordered or not, and then we do mass spec. And in that case, what we see is that so this part is quite stable. These parts they get chopped up within 15 minutes. This one is stable for several hours. So that we normally do. And what we are going to do next is because we are after something like this, or maybe this for the complex. So now what we are trying to do is, because this becomes quite big for NMR, we are actually thinking of doing mass spec hydrolysis. So HXMS. So basically take the protein and then chop them and then put it in U2O and then do the mass spec.
Yeah.
But yes, I mean,
and I'm just
no, these are actually very good suggestions, and again, I mean, not something that we do
and very
mass
simple.
spec. Yes, for a limited purpose is very simple. So once you standardize it, it's very powerful.
Anybody else other than you? Anybody else? Can ask the last question.
So the last question is, sir, this Hawk's protein, in that case scenario, we saw that unfolded region went back to folded region.
Okay.
Yes, that part. As sir told, it's one of the potential reason could be the electrostatics mechanism from the negatively charged beam.
Right.
So is it like, have you seen any other systems in which the IDPs are going back to the folded state? Or is it like some special system of rare?
No, there are many cases. Okay. So I mean, the context changes. So there are many cases where you have a disorder. Once it binds to something, it forms alpha helices, right? Or many times it will not form alpha helices, but it will still be quite ordered when it is bound to other proteins. So those type of sequences are called morphs. MORFs. You will come up with this acronym. I forgot what morphs means. But yeah, so these are long segments which fold once they bind to something. Reverse is also same. So we have studied one protein where there is a helix like this. When it binds to any transforms. So you can get other. It all depends on how the energetics is formed, right? So here we see folding upon binding. In that example, that's also a transcription factor. So when the helix is quite happily there, when the protein binds DNA, the helix.
Yeah, and another thing is, if you look at this picture is false. This picture tells you the helix is stable and solid and it's touching DNA. But what he meant was, but did not show, that this has in equilibrium with another structure where that helix is, so this is it. So you cannot just look, this structure is not correct. Even in the bound state, this is there.
Yes. So there is an equilibrium between these two.
This is going on. And many folded structures, the dynamics is never represented in a beautiful crystal structure. And I think if you look at his NMR structure that he deposits, he cannot deposit one structure. He deposits 20, at least 20 structures. And they're all different. And he claims all the oral kinds.
All of
I had two questions. Maybe one of them is kind of a comment. It's easy to test. Quick way of testing this hypothesis of the helix dipole. You can just take that and in empty simulation you can put an electric field, even without putting it. So it's like modeling that, right? If you put an electric field, whether there is a conformation also. Right. So even just maybe just taking that idea or that part in empty simulation, I just put a field and see if there is any change.
I mean, it's structure. It's easy for you.
No,
it's cheating.
It's
It's kind of a cheating, and also simplify that what is the effect, whether there is an effect of that electrostatics or not. You can isolate that. Okay. So you can isolate and see if the effect of that field or not. Okay. That's one way of doing it.
Okay. So it will tell you that whether this dipole charge interaction.
This part separately, and you run even I'm just saying that even maybe this part can exist in these two conformations, and maybe if you put an electric field, that will adopt that helical shape. That is possible. I don't know whether the rest of the protein is required or not.
Right.
That can also be done. You take the whole protein and put an electric field.
Okay.
Yeah.
Okay.
But
not
the
the
direction of
DNA.
the field might become important. And
Okay.
Yes. But it's possible to add electric field even in Bromax normal solution. So I just thought.
Okay, so
might
just
be a good
take this
idea
and
to-
so don't even need the DNA.
No,
just-
I am modeling the DNA by a field.
By the
so I'm
field.
we're just trying to
I
check
see.
that by just putting an electric field,
Whether this
whether
happens.
the helix forms or
And I'll suggest a very simple experiment. I think there is a big possibility that that little thing will bind phosphate. And it's going to bind phosphate at the end terminal. There are many
okay.
examples, but phosphate binding at the end terminal induces heat.
Okay.
So phosphate binding sites in proteins are often at the end terminal of helices. So if, in addition to helix way, you can get that peptide, right? You can ask a student,
Just do a phosphate titration.
phosphate titration. All you do is you need a CD machine. And if you see that, that would be very interesting. Anyway, we need to see the sequence.
right. And my second question was, do you have a time scale of that tryptophan coming, going in and out? That because your dynamics is, I think, in the few tens of nanoseconds time scale when you're looking at the backbone.
This is
But
so you're
from
talking about
experiments, yeah, from your NMR, you don't get that dynamics.
okay.
Time
So
scale.
there are actually better NMR experiments. We have plans of doing that. We have not been able to do it, which is basically do PRE. So what you do is put some paramagnetic molecule here, and from there we can actually get this time scale, and also that will be much more better experiment than what we have done. So, but
Yeah,
from
another special.
whatever experiment we have done, you cannot get the time scale. From PRE, you can actually.
Anything else? Okay. If not, let's thank Soumya once again. And talk. And join for tea outside.
We work with a protein on students. Do you have work experimental foundation?